science community. We are over 150 organizations from like companies, institutions, and so on. And this webinar in particular is under the Materials and Chemistry Working Group. So I've pasted the link to the YouTube playlist where we have the previous talks. And yeah, we have these webinars every Thursday at the same time. All yours, Hunter.
Awesome. Try to share my screen.
Yes.
Okay, are you seeing the full presentation?
No, not yet.
Ah.
Maybe did you share your full screen?
Yeah, let me try to share the full screen. Not my slides. Sorry, one sec. Pattern in Safari settings for google.com. Okay, sorry, I need to change my settings. 1 second.
Yep. It didn't give you the option to share the full screen?
It did, but then it gave me an error message. How about now? Oh, there we go.
Yes.
Okay. Now I don't know where my slides went. Okay, how about now?
Perfect. Awesome.
Awesome.
Yours.
Great. Yeah, thanks so much for the invitation, and thanks everyone who's on the call for calling in. So yeah, today I'll talk about this paper that we recently presented at ICLR called Unlocking Guidance for Discrete State-Space Diffusion and Flow Models. I'm Hunter, and this work was jointly done by 3 members of the Listgarten lab. So along with myself, this was joint work with Bear, who's a now 4th-year graduate student in the lab, as well as Stefan, who was a postdoc in the lab. So just wanted to thank all of them from the start. And yeah, you know, I haven't attended the seminar series before, but you know, anyone should feel free to stop with questions throughout the talk. This can be as informal as you all like. And I'll try to, you know, make sure that I leave some time at the end for questions, but feel free to stop me. Does that sound okay, Luis?
Perfect.
Great. Okay, so yeah, I'm going to start with kind of some general background of both the motivation for this work and also my personal motivation for working on this problem. And for those of you who don't know, our lab is particularly focused on machine learning methods for protein engineering, although, you know, we fall in this general bucket of kind of AI for science. And when I think about the general framework of AI for science, a lot of problems in this space can be thought of as conditional generation tasks. And so in protein engineering, for example, we're often trying to build a model that can generate different protein sequences, but of course, not just random protein sequences, but protein sequences that have particular properties, such as the ability to degrade plastic, in this example on the left. In small molecule drug discovery, we're trying to generate molecular graphs, but condition on the event that these molecules can bind a particular target with some affinity, specificity, and, you know, various other drug-like properties. And similarly, in synthetic biology or DNA engineering, we might want to design or generate a promoter conditioned on the property that can activate transcription in some cellular context. Now, I'm also going to spend some time kind of with a motivating experimental collaboration that really led to all of the work that I'm going to talk about today. And it has also been kind of, you know, I think provides a nice sort of instantiation of all of this kind of more abstract theoretical stuff that we'll talk about. So in addition to kind of developing machine learning methods, my focus is really on applying these methods to particular protein engineering problems. And for the past 3 years, I've been collaborating with Dave Savage's lab at UC Berkeley, along with a postdoc in his lab, Maria Lukarska. And their lab, one of the things that they are great at doing is building experimental methods for protein engineering. So building really great high throughput assays, and doing all of the wet lab experiments necessary to actually characterize proteins that we might design computationally. And so Maria was really interested in designing a particular class of enzymes called adenine base editors, so ABEs. And these are kind of a new generation of CRISPR technology. So they're proteins that can, in principle, provide very specific changes to someone's DNA or to the genome. And the way they work is by leveraging kind of the original formulation of CRISPR. So for those of you who aren't super familiar, the high-level idea of CRISPR-Cas9 is we have this protein Cas9. It binds a specific RNA molecule that you can program. That RNA molecule will then hybridize with the particular matching location of DNA in a genome that targets this Cas9 molecule to that particular region of the genome. And then what generally happens with CRISPR-Cas9 is the Cas9 will make a double-strand break, and there will be some sort of stochastic mutational outcome that results from that. Often that's some sort of breaking of the gene or inserting some sort of deletion. And obviously in a lot of genetic engineering applications, we want to introduce more specific types of mutations. And so that's where this idea of a base editor comes in. So here, what we do is we deactivate the Cas9 so that it doesn't make a double strand break anymore. And instead, we tether to Cas9 this enzyme called an adenine base editor. And what happens is we retain the ability of Cas9 to localize this base editor to the region of the genome that we want to target. But instead of introducing a double strand break, this base editor will come in and it'll catalyze a specific mutation, in this case from an A to a G. So if the base editor sees a specific A residue, excuse me, in this genomic window, it'll catalyze this mutation from an A to a G. So it can make very precise single mutation edits in the genome. And so, this is a very high potential enzyme that has a lot of great potential applications to DNA engineering, but its properties could be improved for clinical applications. And so in particular, these ABE enzymes, they have pretty weak on-target activity, meaning when we want to use them to edit a particular site in the genome, there are lots of sites where it just doesn't have enough activity. And so in general, we'd like to use protein engineering tools to increase the activity of this enzyme. And then at the same time, these enzymes have some chance of producing off-target mutations, so mutations in regions of the genome that we don't want them to edit. And so we'd also like to reduce these off-target mutations. Okay, so that's just going to be kind of a guiding example of a protein engineering application that we're working towards. And I guess I'll pause real quickly to see if anyone has any questions on that before moving on.
Yep, 1 second. I will give you. Okay, you should be able to unmute.
Yeah, I'm just wondering what values you're looking for and how you determine what values you're hoping to observe or achieve, or whether you're just interested in the variation or deviance or change on its own and where that goes.
I guess here what we're trying to, like, one particular property that we're measuring, and I'll show you some data on in a few slides, is just on-target activity at a particular site. So what we're trying to measure is basically if I introduce this enzyme to target a particular site for a fixed period of time, what percentage of the time does this edit happen? And we can sort of calibrate that based on existing enzymes. and then try to increase that activity further. Does that answer your question?
I think that was a yes.
Okay. Okay, great. Luis, just feel free to stop me if more questions come in. Okay, so when we're working on protein engineering, I like to kind of map it onto this very classic, perhaps overused learn, design, build, test cycle. So what we often do in this machine learning guided protein engineering framework is we start with some generative model. That's this initial learn phase. We'll use that generative model to design a library of protein sequences. Unlike in some chemistry applications, it's pretty easy to make these molecules experimentally. So we just need to synthesize the DNA that codes for these particular proteins. We can then transform that DNA into E. coli, have our E. coli express these proteins, and then Maria has designed these amazing high throughput cell-based selection screens where we can test in one sort of pooled experiment, say 12,000 different proteins. So yeah, let's start with kind of this initial phase, zero-shot generative modeling. I think in the interest of time to get to the actual paper that we're talking about, I won't go into too much detail here. But at a high level, there's two common sets of zero-shot generative models that are used in protein engineering. The first is what we call evolution-based. So these are generally sequence-based generative models that leverage the large amounts of protein sequence data that we have. We can think of all of these protein sequences that we've been able to sequence as sort of instantiations of functional proteins that nature has evolved. And so if we build a generative model to the set of naturally occurring sequences, the likelihood under that generative model tells us something about, say, how likely a mutation is to retain or harm protein function, or say keep a protein folded and at least stable versus destabilize and unfold the protein. Initially, these models were built on sort of family-specific information through multiple sequence alignments, and then there's been a lot of interest in general large language models trained across all protein sequences, such as ESM3 or ProGen. And then a second class of models are structure-based. So these are often called inverse folding models. And here we leverage the large amounts of protein structural data from the protein data bank, and we can train a generative model that conditions on the 3D coordinates of the protein backbone and learns to generate sequences that would fold onto that backbone. So these are models that can leverage this structural information with particularly with AlphaFold 2 and AlphaFold 3, we're able to at least predict the structures of various proteins with quite high accuracy. And so often this structural information can give you sort of more fine-grained information about which sequences are more likely to function or fold on that protein. Okay, and once we've trained these generative models, we can sample from them, construct a library, test them in the lab. Of course, this is actually a pretty hard process. So I've been working with Maria for a number of years, and these assays are quite hard to construct. So initially, I was interested in trying out experiments. So here's just one picture of Maria and I, where Maria was teaching me a little bit about experiments. But it took a ton of work on Maria's end to get these assays up and running. But eventually, we were able to collect this really nice data set. And so what I'm showing here are log enrichment scores. So these are the scores that we get from these pooled experiments where we measure activity by sequencing the DNA of our E. coli before and after challenging them with particularly antibiotic selection, which I haven't gotten into the details of this particular experiment, so that might not make sense. But in the interest of time, maybe I'll just leave it at that, and to say, the scores here are basically measuring a proxy of enzymatic activity. And so what I'm showing here are two replicates of this experiment. and higher scores mean more active enzymes. And in red, I'm showing the initial starting enzyme. And coloring all these points by the number of mutations away from this red wild type. So you can see we get this kind of nice range of activity values. We have a few points that seem much more active than wild type, a lot of points that seem roughly equivalent or sometimes even worse than wild type. But at the end of the day, we have these 2,000 data points that we can now use, hopefully to catalyze further protein engineering.
One question on that. So
Yeah.
you got that data set from this generative model, right?
Yes.
Okay. Got it.
Yeah, sorry. Yeah, so these are 2,000 points sampled from the generative model that we then experimentally characterize in the lab. Thanks for the question. Right. So now, once we've collected this data set, we have this question of, okay, that was sort of one round of this learn, design, build, test cycle. We'd like to kind of keep iterating on this cycle and now leverage this data that we've collected from the first round to further improve the modeling and improve the designs for future rounds. And so the question is exactly how should we do that? And that's really the motivation behind the paper that I'll spend the rest of the time talking about is this discrete guidance approach that I'll talk about is sort of one way that allows you to take some initial generative model that you've trained, say on protein sequences or small molecule graphs, take some predictive model that can predict properties of the molecules that you're interested in, and then leverage that predictive model to guide the sampling of those generative models towards sampling particular molecules that have properties of interest as dictated by your predictive model. Okay, so yeah, that's this paper. And right, so we set up the problem as this goal of trying to sample conditionally from a generative model. So you can think of, we have this initial generative model that I presented before, which might be modeling sort of general base editor sequences. It could be an evolutionary based model, an inverse folding model. Now we have access to some data set, such as the one that we talked about. And we can build a predictive model to say predict from some protein sequence, what is its log enrichment score, what is its activity score. And now we want to sample or generate a conditional distribution of X, so sequences given some property Y, where that property could be something like on-target editing. Okay, there's many ways we could sample from some conditional distribution or construct some conditional generative model. Perhaps the first and most straightforward way we could do this is just to directly train a conditional generative model. So given a bunch of XY pairs, I can train a neural network that takes as input Y and learns to output X. And in this talk, I'll call that a trained conditional generative model. And that can work quite well. So these are commonly used in lots of, say, image applications. They tend to work really well when you have lots of XY pairs. So you can train this generative model on many, many examples. The difficulty is that these X's, so these, in this case, molecules or images, protein sequences, tend to be very high dimensional. And so if we're going to directly train a generative model on them, we need lots of data to really learn that distribution. And so this can be challenging if we don't have a lot of labeled data, as is often the case in the sciences. And so that leads to kind of a second class of approaches for conditional generative modeling, which involves using essentially Bayes' rule. So instead of directly constructing this distribution of X given Y, we can start with some prior distribution on X. So this can be a generative model trained on a bunch of unlabeled examples of X. Then we can bring in a predictive model, P of Y given X. And then Bayes' rule tells us how we can combine these two models to sample from the conditional distribution here. And so there's various methods that have exploited this general idea. And today we'll focus on this second class of methods, which has this particular appealing property that instead of having to directly retrain any sort of conditional generative model, at test time, we can take this predictive model and actually use it to just guide the sampling of our unconditional generative model at test time. And so that's the essential property that these diffusion models have. And so that's why we chose to focus on looking into these diffusion models in the discrete setting here. So I'll kind of introduce diffusion models and this notion of classifier guidance, and then we'll go into kind of how to extend this to discrete spaces, which covers protein sequences, molecular graphs, and these sort of applications. I think there might be a question in the chat.
Yeah, I'm just curious if you can spend even 5 seconds on describing what your axioms were and why, but it's not necessary. It's just my curiosity.
What was?
What your assumptions were, what were your axioms and why? Like what truths did you give it at the outset in your parameters for your sample space?
I'm not sure I understand the question.
Okay, don't worry about it. It's cool. It's cool. I'm sorry to interrupt.
Okay, no worries. Okay, so you know, at this point, many people are probably familiar with diffusion models, so this will be pretty quick. But I'll give kind of a specific overview of diffusion models from one lens, which is this notion of how we treat time in diffusion models. So there's two subclasses of diffusion models. Originally, it was formulated in what we now call the discrete time formulation. And then later it was extended to the continuous time formulation. And this distinction will be particularly important when we think about classifier guidance for discrete state space diffusion models. And so let's talk about diffusion models in continuous spaces, which is how they were kind of originally formalized and certainly originally used for. So for objects in continuous spaces, such as images, where we can model pixel values continuously, as opposed to discrete spaces, such as sequences that take on a discrete vocabulary. So we're working with continuous objects like images. And the idea is we can start with our data of interest, in this case, different images. And we can imagine defining a forward process that adds some Gaussian noise for a discrete number of time steps. So this is what makes it discrete time. If we add enough noise, this image will look like a random sample from a Gaussian distribution. And then what we do is we train a neural network that learns to essentially denoise these images. So we train a neural network that, given some noisy example of an image, tries to predict what the original clean image there is. This is the denoising neural network. Once we've trained that neural network, we can sample some Gaussian noise. We can predict the clean distribution, use the forward process to map back and update a time step in this denoising process, and we can iteratively denoise until we get back to our clean data. So this is now the generative process. So this is the kind of what we do at test time. We sample the noise, we use our denoising neural network to iteratively refine our sample until we get some final example of our clean image. Okay, and then this framework was extended beyond discrete time. So in this discrete time formulation, we had to fix how many times we're going to sort of noise our data. And in this paper by Yang Song and colleagues, the idea is we can instead define a forward stochastic differential equation, which is just continuously adding Gaussian noise through some time interval. We can analytically sample from this, or we can analytically integrate, I should say, this stochastic differential equation to sample an image at any time through this continuous time interval. And then it turns out that we can reverse this stochastic differential equation to go from our noise back to our clean data. And the key object that we need to reverse this stochastic differential equation is this object called the score function. So this is a time-dependent gradient of the log density of our data noised up to a particular period in time. So we can think of noising our data up to this time point across many examples of our data that'll induce some distribution of our data noised up to some time period. And then the gradient of that density of our data noised up to that time is this time-dependent score function. And I think I missed the slide here, but essentially the denoising neural network that we had just here, the magic is that this denoising neural network is actually learning the time-dependent score function. And so by learning that score function through denoising, we can actually reverse the stochastic differential equation and numerically integrate back from our time here, say t equals capital T, all the way back to t equals 0. Okay, so all of that setup becomes important in thinking about conditional generation. So now I'll introduce the method of classifier guidance for these diffusion models. And so now instead of just sampling from some unconditional distribution P of X, we want to sample from the distribution of X given Y. And again, Bayes' rule tells us how we can combine a predictive model, P of Y given X, with that unconditional generative model. The issue is we generally will have this intractable normalizing constant, P of Y, which means we can't exactly compute these likelihoods, and it makes sampling from this distribution difficult. And so this approach introduced by Darwall and Nickel introduced a method called classifier guidance, which sidesteps this issue. So the idea is we want to denoise our data and sample from this distribution. All we need is this object called the score function. If we want to do this conditionally, we can look at this conditional score function of X given Y. And if we plug in Bayes' rule, we'll see that the P of Y term here drops out because the gradient of the log density of P of Y with respect to X is 0. And so the only thing that we need to get this conditional score function are these two separate gradient terms. So the first is the original score function of our data noised up to some time t. This is exactly the object that we've learned from our denoising neural network. And in addition to this term, we need a second term, which is the gradient with respect to X of the log density of Y given X, where we've noised X at some time t. So this is the introduction of our classifier with the additional condition that we need to train this classifier on noisy examples of our data. So we train a time-dependent classifier that, given a noisy example of X, can predict Y. And then we can use automatic differentiation to get the gradient of that classifier with respect to X. Combine these two terms, and now we have this conditional score function of X given Y. So now we can use this to sample the stochastic differential equation and sample examples of our image conditioned on some property Y. And so this is, the key idea behind lots of text-to-image models. So we can generate images conditioned on the property that they correspond to some desired text prompt, such as Technopunk Cats doing molecular biology research. And in the sciences, we've seen lots of great examples of using this classifier guidance approach. So one example is this paper that introduces a method called Chroma, where they first train a unconditional diffusion model that can generate different protein 3D backbone structures. And then at test time, they introduce these classifier guidance to guide the sampling to generate 3D structures of proteins that have particular properties, such as folding into a particular fold class or binding a particular partner. Okay, so that was kind of all of the background about diffusion models in continuous spaces and classifier guidance. And the goal of this work was to extend these approaches to discrete spaces. So in the applications that we're particularly interested in the sciences, we're often learning generative models on discrete objects, such as protein sequences, small molecule graphs, or other molecular complexes. And right, the challenge here is that in discrete spaces, we don't have gradients. And so a lot of this theory and formulation behind diffusion models doesn't directly carry over to discrete objects. And so the first thing we need to sort of understand is, how do we even talk about or define diffusion models on these discrete spaces? And then once we do, how do we extend, or can we extend, properties such as classifier guidance to these models on discrete spaces? And so other people have really led the way of kind of formulating diffusion models on discrete spaces. So I'll kind of review briefly that work, and then we'll jump into sort of our work, which is really tackling the problem of extending classifier guidance on top of these models. Okay, so in continuous spaces, we define diffusion models by adding Gaussian noise to our images or to our protein backbones. When we're talking about discrete spaces, such as protein sequences, so I'm showing an example sequence here, it could also be a small molecule graph. One way to define a diffusion process that iteratively adds noise to this data would be to define a mutation process. So we could imagine in this discrete time formulation, sampling for every time step with some probability p, we will mutate each position in this protein. So we'll go to the first position. In this case, we won't mutate it. We'll go to the second position. This time we'll choose to mutate it. We'll mutate it to a random amino acid, in this case threonine, and we'll do that for every single position of the protein. And then we do that over and over again for many time steps until at the end we get a totally random protein sequence. So that's one way to define diffusion models on discrete spaces. A more commonly used approach is this masking-based noising process. So instead of randomly mutating a position with some probability p, we'll just mask it. So with some probability p, we'll convert the amino acid to this separate token called a mask token. And once an amino acid has been converted to a mask token, it'll stay a mask token throughout the process. And again, we'll do this over and over again until we get a completely masked protein sequence. And so this is very similar to how masked language models work. And in particular, and if we have time, I'll talk about this at the end. There are lots of connections between masked language modeling and these discrete diffusion models with a masking-based noise process. Okay, so that's sort of how you can define diffusion models. You're iteratively adding noise to your discrete data. That's generally either done through a mutational process or a masking-based process. And now let's ask the question of, how do we incorporate classifier guidance and what makes this tricky? So if we want to now, so let me back up. Once we've defined this noising process, we do the same thing that we did earlier, which is we train our denoising neural network to take in some noisy example of this data and try to predict back the original clean data. And so that's this object right here. So given some noisy example of our sequence, can we predict the original clean sequence? And now when we want to sample condition on some property y, we don't have this score function interpretation because we're working in discrete spaces where these gradients don't exist. And so instead we go back to Bayes' rule. And Bayes' rule tells us how we can reverse this diffusion process to sample a less noisy example of X, condition now on some property y. And so the way we do that is we take our unconditional generative model and we combine that with this time-dependent predictive model that can predict y given some noisy example of X. And again, we'll run into this issue that we have this intractable normalizing constant on the bottom. And so just to visualize that, imagine we have a 2 amino acid protein, so a length 2 protein, and it can take on one of 3 states, A, B, or C. In any discrete time interval, we can either not mutate both of these positions, or we could mutate the second position here, A to B, A to C. We can mutate the first position from A to B, A to C, or we can mutate any combination of these positions. And so essentially, we get this combinatorial explosion of the number of states that we could transition to at any time interval, and that makes this normalizing constant intractable. So exactly computing Bayes' rule here is just not tractable. And I saw, is there a question in the chat?
Yeah, just briefly, like what are the A's then to you? And then can you describe in 15 seconds or 30 seconds in words like a story that explains Bayes' rule to people who aren't familiar with it, right? And then what are the assumptions that you're using to get your probability?
Right. So I think you can think of this as like just a protein sequence that has two positions. So a dimension 2 protein sequence, and just for illustration purposes, instead of showing all 20 amino acids, I'm just imagining that we only have 3 amino acids. So what I'm showing here, all the possible protein sequences that you could sample in some particular time interval. And then the second question was, I guess, about Bayes' rule for people who might be less familiar. I guess let me think about that for a moment. I think essentially what Bayes' rule is saying is, if you want to sample some X condition on some property Y, you can think about sampling from your unconditional distribution X, but now weighting all of those samples by the probability that those samples have the desired property Y. So Bayes' rule is essentially this reweighting term that's saying, you can kind of think in your head of an algorithm that's just sampling unconditionally from some distribution and then weighting them by your property of interest and then resampling there.
Right. So what's your property of interest, or what is the thing that you're looking for?
Yeah. So in this case, that property of interest could be, so your X's would be protein sequences, and your property of interest could be on-target editing efficiency of these protein sequences at your particular site in the genome for this base editor application. Or it could be something simpler, like just how stable is that protein? Or if it's a molecule, maybe how tightly does that molecule bind a particular protein? Does that help?
Yeah, thank you very much for your time. I appreciate it. Thank you.
Yeah. Okay. And so, right. So it seems right now that computing Bayes' rule here is intractable. We have this combinatorial explosion of the number of states that we need to consider. So in this 2 amino acid system, that's only 9, but essentially this scales exponentially with the dimension of the object that you're modeling. So as we go to larger and larger proteins, this becomes completely intractable. And the key insight for this work was, well, even though we have this exponential number of states that we need to consider when computing Bayes' rule, we don't actually mutate to these different states with equal probability. So I described this mutational process that happens where we mutate each position in this forward noising process with some probability P. And that probability is basically proportional to how fine-grained our time steps are. And so in this setting, we will, in any time interval between t and delta t, we'll stay in our current state with probability 1 minus p squared. So that's the probability that we don't make a position, we don't make a mutation at this position, and then we don't make a mutation at this position. Similarly, we'll make a single mutation with probability p 1 minus p, and then we'll make 2 mutations with probability p squared. And so if p is very small, we'll be more likely to stay exactly where we are in any time interval. And similarly, we'll be slightly less likely to make a single mutation. And then we'll be even less likely to make more than 1 mutation, so any sort of double mutation. Is there a question?
Question, yes.
Hi, everyone. Can folks hear me okay?
Yeah.
Perfect. So just to clarify then, there's no conditioning of this mutational probability on anything physical. So for instance, like if you were to throw like a proline in the middle of an alpha helix, right? That's probably not a condition that you'd see occur naturally in nature. So there's no conditioning on like these physical priors, I should say.
I guess that depends on your generative model. So I guess that there's a few ways to answer that, but one is, the unconditional generative model should probably learn not to put prolines in that alpha helix if it's been trained on enough protein sequences, right? So if these are trained on naturally occurring protein sequences, the unconditional generative model itself will pick up the pattern that you can't put prolines in the middle of an alpha helix. Or if you train this diffusion model as a inverse folding model where it conditions on the 3D structure of the protein, then it'll definitely, easily learn not to put something like a proline in the middle of the alpha helix. So it's not we don't bake in those rules, but they're implicitly learned from the data. And the better the generative model, the more likely it should be able to learn those types of rules.
I see. Okay, so you're working with an already like pre-trained generative model here. This is not like training from scratch.
Yeah. So I guess both. I think, I could have set this up more clearly. I think now that we're talking about classifier guidance, you can think that I've pre-trained some generative model, and now I just want to sample from this conditional distribution of x given y. And so I want to bring in a separate classifier and guide the sampling. And I'll mention some of the generative models that we use, but you can think of this generative model as a pre-trained inverse folding model that's just trained under this diffusion framework to generate different examples of protein sequences given a 3D backbone structure.
I see. Perfect. Thank you.
Sure. Okay, so we have this exponential number of states that we need to consider for computing Bayes' rule, but we realize that we don't actually mutate, or we don't sample these states with equal probability. And in particular, in this discrete time formulation of diffusion models that I've set up, we're free to decide how fine-grained we want to chunk up this discrete time interval. So imagine I have some time interval between 0 and 1. I get to choose how many time steps I want to take. That just increases the computational cost, but I'm free to choose that time interval, delta t, and make it as small as possible. And so now imagine I make that time interval smaller and smaller. As I do that, what I'll see is I'm now increasing the chance that I either stay exactly where I am or just make a single mutation, and the probability of making more mutations decreases faster because it goes up with the exponential in the number of mutations that I'm going to make. And so as we take tinier and tinier time steps, we're essentially going to always either stay exactly where we are or make a single mutation. And that's kind of the key guiding intuition for why guidance might be tractable in these discrete spaces. I know that's a little abstract, but we'll get there in a second. And so this motivated kind of the final kind of object that we need for making guidance work here is to, just as in the original formulation of diffusion models that I talked about earlier, we started with this discrete time formulation and then moved on to this continuous time formulation where we defined a stochastic differential equation that noised our data, and we reversed that stochastic differential equation. These set of papers from Campbell et al. Introduced continuous time formulations of diffusion and another class of models called flow models on discrete spaces. And the idea is we can define our mutational noising process instead of through discrete time steps. We can continuously define that noising process through an object called a continuous time Markov chain, which might be familiar to those of you with a chemistry background. So the idea here is we, instead of directly defining these transition probabilities through some time interval delta t, we define continuous transition probabilities through this object called a rate matrix, which describes the instantaneous probability of transitioning from some state x to some other state x tilde. And then we can sample this continuous time Markov chain through a continuous time interval. And so essentially most of the time, we'll just stay exactly where we are in our current state. But eventually, we'll transition out of our current state x to some other state x tilde. So we'll jump, and that'll happen based on the relative rates between all of the different x tildes that we could transition to. And then we'll hold and stay in that current state through some continuous time interval, and then sample a jump, and we'll do this over and over again. So this describes kind of the dynamics of this continuous time Markov chain, which we'll use to define kind of a mutational process on this protein sequence. So here we're going to start with our clean protein sequence. We're going to define a forward noising process where instead of predefining the time intervals at which we'll sample noise, we'll use this continuous time Markov chain to decide at what time do we transition out of our current state, make a mutation. We'll sample that, in this case, T1, and we'll do this over and over again until we reach the desired time, T equals 1. So we parameterize this forward noising process through a rate matrix. We can numerically integrate the CTMC and sample this forward process. And then we can train our denoising neural network just as before to go from some noise protein sequence back to the original protein sequence. Once we've trained that denoising neural network, we can actually use that to compute the reverse rate matrix that's shown here, which defines a CTMC that takes us from this completely noise protein sequence back to the clean protein sequence. So at a high level, we're using this denoising neural network. We're using that to get a reverse rate matrix, which defines a new continuous time Markov chain that brings us back from our noisy sequence back to our clean one.
There is one question.
Sure.
It seems to me like your interest, right, is like how many steps removed from the original can we go while trusting the AI brain, right? And then knowing that many steps removed from your ideal or whatever, or however that's captured, will allow you to be like, we can trust this many steps of reverse engineering as likely valid. But I'm curious, like, what your ultimate goal with that is, like, just to be able to trust the AI for a certain number of steps, or like improving that the original is the original, or how you see the idea applied, if I'm describing it correctly.
I think maybe there's a different way to think about it, which is the reason we're talking about all of this kind of like noising and denoising process is actually it's just a way to learn a distribution over our protein sequences. So the goal here with these diffusion models is to learn based on some set of existing protein sequences, sort of what is the underlying probability distribution that explains these sequences. And so such a distribution would be able to learn what types of mutations are tolerated in a protein versus not tolerated. Or in small molecules, it might be what sorts of molecular graphs are reasonable synthesizable molecules.
And are you saying it to be tolerated, it has to maintain an A, or you're interested only in B and C, and like how often that arises and at what time point?
So the distribution that we're ultimately trying to learn is just this clean time point at T equals 0. And we're interested in the what is the general composition of A, B's, and C's that are allowed for that we see in naturally existing proteins. And the reason we're using all of these kind of what you can think of as latent noised versions of this data is just that gives us a way to instantiate an algorithm where we can sample just a completely random protein sequence that won't look like a functional normal protein at all. It won't look like an example from the distribution we're trying to model. But if we train this neural network correctly, we can start with this completely uniform random sample of a sequence and iteratively clean it up until we get to a sample from our desired distribution.
I'm not sold, but I will. I thank you for your time. I've been answering the question and being receptive to it. Thank you.
Sure.
One quick question. So if I remember correctly from the paper, this CTMC, one of the properties is that you do only one change at a time, right? Like only one amino acid change.
Exactly.
And that's something favorable. I don't remember exactly why. Can you like?
Yes. Yeah. So let me jump to that. So basically, once we've trained this denoising neural network, we can sample the CTMC, and so we'll start with our completely noise sequence. We'll denoise it, and we'll iteratively denoise it until we get back to our original sequence at t equals 0. And this is exactly the point that you just mentioned. The idea is in this continuous time formulation, every transition will only mutate a single position at one point in time. And so intuitively, in the interest of time, I think I'll go kind of quick over this, but intuitively, the CTMC is defined per each dimension. And so you have like, in this case, 5 parallel CTMCs defining the denoising process for each of these positions. And for each CTMC, you can imagine sampling what is the time at which I transition each of these positions. And because we're talking about continuous time, there's 0 probability that any of these positions will denoise at exactly the same instantaneous time point. And so you always have, you'll always have a shortest time among all these positions. And at that time, there will be a single position that you unmask. And then you'll denoise again and sample the time at which you denoise each of these positions, take the shortest one, and you'll denoise the next position. And so at every single position, you'll denoise or you unmutate or unmask one position at a time. And that's just exactly this intuition that we started with before, where we said as we decrease the time interval between any 2 steps, the probability that we make more than one mutation gets smaller and smaller and smaller. And so in this continuous time limit, where our time intervals are infinitely small, we have exactly 0 probability of making more than one mutation, and we'll only either stay exactly where we are or mutate one position and not the other. And okay, so that's kind of the intuition of how that happens. Why was that important was the second part of your question. And that becomes important when now computing Bayes' rule. So before we had this intractable normalizing constant where our space of possible transitions blew up exponentially. But if in the continuous time setting, we're only ever making a single mutation in any time step, then instead of having an exponential number of states, we'll have just a linear number of states. So these are all the states where we either make 0 mutations or just 1 mutation. And this makes computing this normalizing constant in Bayes' rule completely tractable, because we just have a linear number of states instead of an exponential. And that's kind of, now I know we're up on time, but this is basically the final kind of punchline of the paper is in continuous time, Bayes' rule becomes tractable to compute, which means we can define analogous classifier guidance and a related class of methods that I didn't talk about today called classifier-free guidance. We can define analogous equations which have been formulated for continuous state space diffusion models. And we have our own set of now tractable exact equations for classifier guidance that we can use for discrete state space diffusion models. So here you take your original unconditional generative model defined through this rate matrix, and you modulate the rates, in this case by this likelihood ratio. So if we're transitioning from x to x tilde, we're going to increase the probability of making those transitions if x tilde is more likely to have the property y than x. And similarly, we'll decrease the rates if x tilde is less likely to have that property. But this is all, kind of mathematically derived in the paper. And then I'm sorry that I don't really have time to maybe get into these experiments too much, but the application here was to really use these methods for different scientific applications, such as conditional generation of protein sequences, conditioned on, say, the property that they're stable, or in small molecules, we had a small example where we generated different molecules condition on various, kind of toy properties like the number of rings of these molecules or the log P, lipophilicity of these molecules. But I think in the interest of time, I'm going to kind of skip over this and just kind of wrap up and say, so what we showed here is there's this method of classifier guidance in continuous space diffusion models that allows you to take some pre-trained diffusion model, guide it at test time with a predictive model, like a classifier, to generate from or to sample from a conditional distribution of your molecules given some property Y. And we can extend that method of classifier guidance to generative models on discrete spaces. And these allow you to build conditional generative models over, say, protein sequences, text, small molecule graphs, all of these discrete objects. And so I'll wrap it up here and take questions for the time that people have.
Yep, there is a question.
Yeah, that was a really great talk, Hunter. I really enjoyed your explanation of Bayes' rule. That was really clean, I thought. Is there any argument that you can see for using like non-Markovian dynamics in this process? Because it seems like the time step is really like your critical parameter here. And it seems like using like a memoried transition would help you compute that time step a little bit more rigorously.
Yeah, that's really interesting. I hadn't thought about that before. And so I don't know that I have anything insightful to say to that question. But I guess as I'm thinking about it, it does feel like, there's definitely potential that could be helpful. In particular, like if your denoising neural network kind of knew about previous states, that might help it understand kind of how to better unmask the current state. Yeah, I know like one area people are really interested in with these discrete diffusion models is how to speed up sampling so that you don't need to take as many time steps. And it's possible that these kind of like a non-Markovian formulation of these noising and denoising processes might help with that too. So yeah, I think that's an interesting idea. I'm sorry I don't have anything very insightful to say about it though.
No, thank you.
So last chance for any questions before we wrap up. Okay, I think everything was super clear. Again, thank you so much for presenting. And you can find more details in the paper itself, and you can contact him, I guess, if you have more questions. Thank you everyone for showing up. And again, thank you, Hunter.
Yeah, thanks everyone for coming, and thanks Luis for the invitation.
Of course. All right, bye everyone.
Bye.